Good afternoon to all, and I hope all of you are not sleeping. Yeah, wake up. So, as you people all introduced yourself in the morning, there's so many botany students and zoology. So how many students have done molecular biology experiments, DNA work? Can you please raise your hands? Oh, very few? Okay. Okay. So, I think for others, this might be a slightly new topic. But I'll be just talking some basic things so that'll be easy for you all to understand. Today, I'm going to talk about the role of digital sequence information in biodiversity conservation. Now, you've been hearing from morning about uses of biological resources per se for the access and benefit sharing mechanism. I think we already heard 3 talks on access and benefit sharing mechanism, Biological Diversity Act. So you've been hearing about plant parts being used and how benefit sharing system is working for plants, animals, microorganisms. But now, if you see the technology, we are far ahead. So more than this, if you see now, there are studies, lot of studies going on at the molecular level or at the gene level. So, but at least all of you must have studied in your college about DNA, RNA, right? So can anybody say the full form of DNA? Okay, and RNA? Okay, so thank you. So here, this is, you know that DNA, RNA and the proteins are all the genetic structure of any biological resource. So ideally, it is also part of the biological resource. So if you see, the flow is, you have the DNA at the supreme, and then it translates information into RNA, which then translates into amino acids or proteins. And that's then you have the reaction that happens in a body or an organism. But now why am I saying this? I'll connect you that slowly. But before that, first, so now as I told you, the flow of information is the genetic resource that is actually the metadata. That is, the DNA sequence has the whole information of a particular organism, then which is the genomics, or that's how we call it, the study of genes, which then when you work on RNA, it is called transcriptomics, because then you're translating the sequence or the information from the DNA into an RNA. And then comes the protein, which is again proteomics, and which then develops some metabolites or anything, some products or derivatives in an organism for the organism to be functional. So now why am I saying this? Is because, as I already said, this genetic structure or the genetic genes or DNA are also part of the biological resource and should be involved in this access and benefit sharing mechanism. So, but then, how do we see this? Now, when you see about, first, let's go to the very simple mechanism of how do you do this, get this DNA sequences, before I get into the detail of ABS. Now, if you get into any lab, maybe I heard some people were working as a JRF or doing a PhD. So they would be doing a simple research on any sequences. That is, first, you actually have to access the biological resource of interest. Then you do the sequencing, or you isolate the DNA out of it, and then you generate the sequence of it. If you see here, it is this will be how a sequence will be. If you see here, like how, so I hope you know what DNA is made up of. Yeah, anybody? What is DNA made up of? What are the components of DNA? ATGC? Thanks. So you develop the sequence, and then what you do? Now when you want to do a research paper or you publish, the first thing that you have to do is submit the sequence to a database. And that's when you get something called an accession number, which you then use it for your publications. So this is your basic thing that you do for any sequencing. So what we do is, that is, first you develop the sequences of the plant resource that you're interested in, submit it in a database, which I will come in the due course. And then after that, using your sequences, you can then analyze the information that is there or what you require. But then where do you submit the sequences? You cannot submit it in a repository or in a BSI, ZSI. So there are recognized repositories or the databases that actually holds the sequences and which is freely accessible. It's open access. And the three major databases globally are the NCBI, which is the National Center for Biotechnology Information, which is situated in USA. The second is the European Bioinformatics Institute, EMBL, which is in Switzerland. And the third is the DNA Data Bank of Japan, which is in Japan. So now, if any researcher in any country, when they develop a sequence, first submit the sequence to one of these databases to get an accession. That's the first step. So to do a publication or to do any further study. So once they submit that, they then as I already told you, they have a free access of any sequences in the database there. So then you can do your next study, which is you use your sequences, compare with other sequences, and see what is interesting, do your research. So that is, you're accessing sequences from a database of anybody's work. So it's not that you're accessing only your sequence, but you're accessing any sequence from USA or from Sweden or any country. So you're actually accessing sequence, which is you're accessing the genetic resource of a particular biological resource. You're accessing it. So keep that word in mind. And you will be using it for your research. So now you know that there's a database and your sequences. So now if you can actually then differentiate the use of these sequences into different major categories. One is for your research perspective. Second is for commercialization. Now when I see for research, I could identify an unknown microorganism or a plant or a pathogen in a particular organism using the sequences, or when I compare with other sequences. And I can do DNA barcoding using sequences and compare. And if you see, you can do something like a phylogenetic tree where I can identify if a species that I have is unknown. But when I compare it with sequences of other species or in the database, I will know what it is. So you can do identification, you can do taxonomic work, or it's called molecular taxonomy. And all this can be done, is all non-commercial research, which is publication or you submit in a conference paper. It's all non-commercial research. But now next, you can use this digital sequence information for commercial perspective. That is, you can develop transgenic crops, you can develop high-value desirable molecules, metabolites. It can be used in medicinal research, you develop products. And if you see vaccine development, if you see in COVID or Ebola vaccine has been developed using these sequences. So there's a lot of commercial potential also in the use of digital sequence information. It's just that how you use it matters a lot here. So now, if you see, now I would like to connect this, the use or the access of digital sequence information to the convention, which you have been hearing from morning, that is the Convention on Biological Diversity. It's just a repetition here. The three principles of the CBD is conservation of biological diversity, the sustainable use of biological diversity, and the third is the fair and equitable sharing of benefits arising from the utilization of this biological diversity. Now how do I put this digital sequence information in these three slots? So before that, Now I have to see how do I see digital sequence information globally or in the context of CBD. If till date, till now we had our COP conference of parties 16, which happened very recently. Till now, this digital sequence information has not been defined. It's just that it has a placeholder name in the decision saying that it refers to the data derived from dematerialized genetic resources. That is, now I know genetic resource can also be a biological resource which you access, but once you take the information out of it, it is dematerialized. I don't need to access the resource again. It's just I'm going to access the information. So it is a dematerialized information, and that is how CBD has till now mentioned it as the placeholder. Now, how do I see this? So that is, like how I said, you see the sequence data as the material information, or the protein data, or this. Now when I said, because it's a placeholder, now how do I see it in the three conventions? So if you see the last point here, it relates to both 1 and 2. Conservation, that is, you can do your research using the sequences to see how you can conserve a particular bioresource. That is, you can do something like a population genetics. You identify which are the species which are actually very susceptible. You try to conserve it, you try to restore it, and also see what is, I mean, when you talk about bioprospecting, you can identify which is the species that has high value and see how can it be sustainably used. So there is a lot of potential in using the first and the second step. But now here I am going to focus on the third principle, that is the access and benefit sharing mechanism. Now how do I get the access or the benefit sharing from the access of DSI? Is here. Actually, that's a big $1 million question which is still not been decided. Now, but now before that, I told you that it has been kept as a placeholder, not defined. So there is a school of thoughts that DSI can still not be decided that it is part of the convention or it can be included in the convention. But if you see the Article 15 of the Convention of the Biological Diversity Act, Biological Diversity, it says access 15.4, if you see, the access where granted shall be on. This is, okay, now here, sorry. Now here, when I'm talking about access and benefit sharing, like how Dr. Suhas talked and Shri Goyal sir talked, we talked about two things. When you are accessing a biological resource, the two important things is the prior informed consent and the mutually agreed terms. That is how we sign an agreement for any biological resource for the access of it. But now here is the data that is found publicly available globally. Now how do I sign a PIC or a MAT with a person who's going to actually access the sequence that I have submitted in a database? It's no point. Even if you see the databases which I mentioned, the NCBI database, 89% of the database does not have the country of origin or where actually the sequence is accessed from or the bioresources from. So the difficult part is identifying who the user, who the submitter is, where the genetic resource has come from. So then how do we sign this PIC and MAT? It's a question. The second, like how I said, prior informed consent and the MAT. Now, I also want to show you how this digital sequence information can be put in the CBD and the Nagoya protocol, which you've been hearing from morning. So if you see DSI, if you see Article 2, you can say that DSI is part of the definition of the genetic resources. That is, if you read out the definition, it says means genetic material of actual or potential value and genetic material that contains functional units of heredity. So which means it's the DNA or the RNA we taken. Same thing, it has also been highlighted in the Nagoya Protocol Article 2C and Article 2D. So now we can make sure that we can discuss about the access and benefit sharing mechanism for the digital sequence information. If you see, even in the Indian context, the Biological Diversity Act Amendment 2023, you see the section 2C, where the definition of biological resources, it says it includes plants, animals, microorganisms, or parts of their genetic material and derivatives. So which means DSI is also included in our legislation. So next, why is this DSI critical within the CBD? Like how I mentioned, it is actually a high technology. I mean, we are in the technology world, and a lot of research has been going on using these sequences, genetic data. But now, how do I get this access and benefit sharing mechanism involved? So what are the challenges? I already said we have the challenge of this brick and mat. And there are some countries which are still not parties to the CBD. For example, it's the United States of America. Now, I just mentioned a few minutes back, the major database center that holds almost 90% of the sequences is NCBI, which is in USA, which is a non-party to the CBD. So now how do we think about the access and benefit sharing mechanism for the sequences that are being held by a country that is not part of the CBD? So, like which I already said, if you see the three databases, the other important part of this is these three databases were actually formed or established before the convention itself came in. For example, this came in 1980s. For example, NCBI came in 1980s. So their mandates were totally different. So there it is just that they are a deposition, and they are the ones who will take care of the sequences. So now how do we get this new concept of access and benefit sharing into their mandate? That's a difficult question. And as I already told you, the NCBI in USA, so why will they agree to get into this access and benefit sharing mechanism? Now, when I see, the next is what is the usage of DSI? Like how I said, now there's so much of technology there, a lot of work is going on using sequences, DNA, RNA, a lot of development, product development and all that. So there have been many studies. If you see the graph here on the right, here, this which has a lot of circles in it, it was a study done on the DSI or the digital sequences information, the sequences that have been submitted by the Malaysia or the researchers in Malaysia in the NCBI. So this is based on the data of the Malaysian sequences. Now, if you see the circles here, all in different colors, they are the quantity of sequences being used by different countries in the world. So if you see here, there's so many countries that are actually accessing the Malaysian sequences and using them so we don't even know if they are taking prior informed consent from the Malaysia. And if you see the larger the circles, more is the data being accessed. So you can imagine how much of data is being used or accessed without we knowing. Similarly, now if you compare the sequences being used and provided by a country, if you see in the highlighted here is India. Now here this is a graph about, if you see the blue bar is the one who provides the sequences to the database. The purple is one who uses the sequence, and the red part is for the non-commercial research. So now if you see India, we are more or less equally doing it. We are also using it, accessing it, and also mainly for the publication. So now when I compare globally, what is the percentage of use of DSI by India? If you see, India uses 6% of the digital sequence information in the databases globally. But actually, we submit only 3.46% to the database. Now, when I think about access and benefit sharing, if I think about in the national context, I will say if you're actually accessing it, then you have to give me benefit sharing. But now if you see, it's almost double. India is using more than actually accessing it. So should India be paying more, paying the benefit sharing more than actually getting? So that's a difficult question. So that is why, and obviously there are other countries who will also say no to this. So in this regard, because there's so much of complexity in this access of benefit sharing of the digital sequence information, this has been going on for past 20 years, or no, 3 COPs, where there was no consensus, there was a lot of debate, deliberations, which happened in COP 14, 15, and 16. So in 14 is when they started asking the question, how do we get DSI? So where their major concern was about the traceability. That is, like how I already told you. Now I submit from India a sequence into the database, but I do not say where it has come from. I do not say the country. Now a person sitting in New York would be using my data. He will not know where the sequence is from. So how can he trace back saying this is a sequence from India? So that way, the database, for example, NCBI, 89% of the data does not have the country of origin, from where it comes. But recently, maybe in the past 2 years, they have somehow said it is a mandate that you have to give the country of origin when you are submitting your data. But that is just 2 years back. But we are having data, billions of sequences from 1980s. So what about that? So that was why there was no consensus in COP 14. In COP 14, there was a decision saying, okay, now we cannot get into a bilateral. We'll get into a multilateral system. So that I don't have to think about traceability at all. It's just that it'll be in a multilateral, many countries will be involved. So whoever uses it can put their benefit sharing into a common fund or the global fund. So that is how it was decided, but the modalities, how to do everything was decided in COP 16 only. So, and as you know, this kunming-montreal global biodiversity framework, the digital sequence information has also been aligned to this kunming-montreal benefit sharing based on target 13. So now the main decision that happened in COP 16 related to DSI is that now that they have established that we will have a multilateral mechanism and a global fund called the Kali Fund, because the meeting happened in Kali. So a Kali Fund has been established exclusively for the benefit sharing that they will receive from the use of DSI here. So this is, and the modalities that were discussed in COP 16 was only for the multilateral mechanism. But there were countries. So before that, I will just tell you like how I told you in COP 15, they decided we should have a multilateral mechanism. So then there were a lot of intersessional work. So there was a working group that was developed by the CBD, having all the parties to be involved and reach to a consensus. They had their meetings, and that's when we came up to a decision 16/2 in the COP 16. So the major problems that, as I already told you, was the terminology. What will DSI, now how do I define DSI? There could be some countries that say only DNA is part of my DSI. There are some countries that says DSI will be DNA, RNA, and protein. And there's some group of people who say DSI is DNA, RNA, protein, metabolites. So that is why there's still no consensus. And India in COP 16 actually really fought to have some kind of a bilateral mechanism also in place with also this multilateral mechanism. They didn't say that we don't want multilateral, but they said now that when I know where the sequence is from or the country of origin, we prefer a bilateral mechanism over the multilateral mechanism. And but still there is always an issue of the traceability of the sequences. For example, if you see, now when I have to develop a product, I will not use only one sequence or two sequences. Or when I do an analysis, for example, maybe few of you know what is a blast. where I try to compare with hundreds of sequences that are already in database and then develop a sequence of my interest and go about in developing a product. So now how do I know where actually the sequence that I have developed based on all these hundreds of sequences is from? So how do I trace it back? How do I cannot pinpoint saying it is only from the sequence. So there's a big issue of traceability, and most of them will not have these geographical areas, and there's a lot of redundancy in the data, so it's difficult to trace it. So based on all these problems, there are a lot of publications you might go and also see on the policy issue, how to go about with multilateral mechanism, there was a study in South Africa, how can they see this mechanism being operated. There are other papers. So now, as I said, in COP16, they went up to saying, we had actually a lot of meetings. If you see in COP16, there were like 12 contact group meetings, presidential meetings, then we had a plenary meeting, and which ended at in the morning, 3 o'clock in the morning of the next day. And that's when it was decided and this decision was adopted on the digital sequence information. So where I already said the major decision was on the fund or the establishment of the SCALI fund and the modalities. Now I will just give you a brief on what was discussed or decided on the modalities of the multilateral mechanism. Now what they did was they categorized them into 5 different things. Now first thing is they ruled out about non-commercial research. They said you are free to access how much of your sequences you want for non-commercial research, for publications and all that. We are not going to get into this access and benefit sharing for it. So this is going to be only for commercial utilization. So if you are accessing any sequences for a commercial motive and you develop a product and you commercialize, you are supposed to share the benefits out of it to the multilateral mechanism, that is to the KALI fund. So which means you will not find any traceability issue in this. So that is, now if you see in monetary contribution, they said an indicative value, all this is still work under progress. They're still in the process of developing committees to actually come to a concrete conclusion. But the indicative here is that they have, or the companies who commercialize a product using the DSI will have to give 1% of their profits or 0.1% of their revenue to the KALI fund. This is now here the monetary contribution, and in non-monetary, as already Dr. Suhas and Goel sir had talked, it's about capacity building and infrastructure development, the use of that's how the non-monetary contribution is. But now next is on difficult task is on the data governance. Now, in the data governance, what they did was, now how do we find out? First, they said the entities that operate these databases should first tell who all are the submitters of the sequence to provide, first to comply with their ABS regulation. That is, for example, if you take our national legislation, I, as a company in India, would like to commercialize a product using a DSI. I have to first, as Sir already said, I have to comply with the national law. I show that, or I prove that I have complied to the database that I have complied, and now you can accept my sequences. So that was one important thing. Second, I said that they want all the submitters of the data, whoever is trying to submit their sequences, should provide the country of origin. And now the third part is the parties that fund the sequence databases. That is, now if you see DDBJ, that is the Japan. Japan is party to CBD. Even European Union is party to CBD. So in that case, those countries have to ensure that all the entities that are operating the databases should ensure that there is this implementation of the multilateral mechanism. That is, they have to inform those people who are submitting the data to be aware that they have these ABS obligations in them. They have to follow that. So the last part was the parties and non-parties are invited to take administrative policies or legislative measures to align with this multilateral mechanism. For example, now the countries like India already has a national legislation. So they are now asking the parties to align with this multilateral mechanism so that there is facilitation for the people who want to opt for the multilateral mechanism over the bilateral. Now then next part is, now we have got funds assumptions. It's because all this is still in a very initial stages. So now assume that we have funds in the Kali fund. Now how do I allocate it? To which country or which party should I allocate it? So there it was again discussed and decided that we will take 3 major criteria to decide how I should allocate, how the CBD can allocate the funds. One is based on the biodiversity richness of that particular country, or if that is of the species for the sequences that they are submitted are endemic or endangered from a particular country, then that amount or that fund should go to that country or the party. Second is the geographical origin is known, and we don't have any other, like for example, red sanders, it is only in India. So if the geographical origin is known, it should come back to India. And third is for use for capacity building and other things. So this is now how it has been going to be allocated. Now how do I disperse it? So there was actually a lot of discussions on the disbursement, and India also put in their strong footing and said that we also have our own sovereign rights over the biological resource that are being used. So, and we respect the indigenous people, the local communities there. So we as a country should decide the amount, how it should be utilized. Excuse me. So that's why the decision got this sentence that based on the national circumstances and national legislation, the funding of the global fund will go to the indigenous people, local communities based on their needs. So now, this is just in a nutshell what all I said, but all this decision and the establishment of Kali Fund is still at an infant stage. Sorry, and there's still a lot of deliberations going on in CBD. So the take-home message here is that one is there is still not a defined, digital sequence information is not defined and it's a placeholder, but has a context in the access and benefit sharing mechanism. And third is the establishment of the Kali Fund. This is for the multilateral mechanism. Thank you.